Hey everyone, let's dive right into this explainer and zoom all the way down into the microscopic architecture of how our brains actually build and store memories. We all know we learn from experience, right? But today, we're going to teach us in depth and look at the exact cellular blueprints that make that learning possible. To really unpack this, here is our agenda. First, we'll look at wiring the brain. Second, the molecular calcium switch. Third, remodeling with AMPA receptors. Fourth, synaptic homeostasis and balance. And finally, making memories permanent.
All right, part 1, plasticity, wiring the brain.
So, the absolute foundation of learning happens right at the synapses, particularly in regions like the hippocampus. This is the realm of synaptic plasticity, which is just the brain's incredible ability to modify the strength of connections between neurons. When we talk about strengthening a memory, we're really talking about long-term potentiation, or LTP. This has been extensively studied at the Schaffer collateral synapses on CA1 pyramidal neurons. Basically, if a synapse gets a tetanus, which is just a brief burst of high-frequency electrical stimulation, it triggers a persistent, long-lasting increase in the size of the excitatory postsynaptic potential. The connection literally gets stronger.
And this brilliantly illustrates the famous Hebbian rule. For LTP to happen, there has to be a correlation. The presynaptic axon sending the signal absolutely must be active at the exact same time the postsynaptic neuron receiving the signal is strongly depolarized. It's the ultimate biological team-up. They literally have to act together to strengthen the bond. Neurons that fire together, wire together.
Now, what's truly crazy about LTP is how wildly precise it is. It is not some messy, random process. First, we have input specificity. If a neuron has 1,000 synapses, only the specific ones that are active get strengthened. The neighboring inactive ones stay exactly as they were. Second is cooperativity. A single synapse usually isn't strong enough to cause this change by itself. Multiple synapses actually have to work together in space and time to strongly depolarize the membrane. And third, longevity. This isn't a fleeting change. LTP can last for weeks or, in awake animals, literally an entire lifetime.
But think about it. The brain can't just keep strengthening and strengthening forever, right? That brings us to the functional opposite, long-term depression, or LTD. In the CA1 region, this is induced by low-frequency stimulation. Think of a slow, steady pulse of about 1 to 5 Hz, which causes a long-lasting decrease in synaptic effectiveness.
So the crucial point here is summarized beautifully by the BCM theory, named after Bienenstock, Cooper, and Manchuro. They basically extended Hebb's rule to account for this bidirectional modification. If presynaptic activity correlates with only a weak or modest depolarization of the receiving neuron, that synapse is weakened. Simply put, neurons that fire out of sync lose their link. This actually happens all over the brain. For instance, in the cerebellum, LTD occurs at parallel fiber synapses when they fire at the exact same time as really powerful climbing fiber inputs.
Moving right along to part 2, the molecular calcium switch.
So how on earth does a neuron actually know whether to strengthen or weaken a connection? Well, it relies on this brilliant molecular coincidence detector called the NMDA receptor. These are glutamate-gated channels, but they have a really unique quirk, a magnesium block sitting right inside the pore. For the receptor to open up, two things must happen simultaneously. Presynaptic glutamate must bind to the receptor, and the postsynaptic membrane must be depolarized. Only when both happen does that magnesium block pop out of the pore, allowing calcium to flood into the dendrite. It is literally the perfect molecular coincidence detector.
And calcium is absolutely the ultimate molecular switch here. The decision between LTP and LTD depends entirely on the magnitude and pattern of that calcium entry. If there's a massive flood of calcium from a strong depolarization, it activates protein kinases, specifically PKC and KMK2. These kinases are the builders. But if there's only a weak depolarization, the magnesium block is only partially relieved. You just get a modest trickle of calcium. This lower level activates protein phosphatases instead, which act as the teardown crew.
Okay, part 3, remodeling with AMPA receptors. Now that the switch is flipped, either toward building up or tearing down, let's look at the actual physical remodeling of the synapse. The physical changes involve moving another type of receptor, called AMPA receptors, in and out of the membrane. Think of the PSD95 protein scaffold like an empty egg carton and the AMPA receptors as the eggs. During LTP, the kinases phosphorylate existing AMPA receptors to make them conduct more current, and they insert brand new eggs, specifically AMPA receptors with the GluR1 subunit, into the carton. During LTD, the phosphatases do the exact reverse. They dephosphorylate the receptors and pluck those eggs right out of the carton, internalizing them completely away from the synapse.
These chemical cascades literally change your brain's architecture. LTP doesn't just stop at adding receptors. It triggers structural plasticity, actually growing and budding brand new dendritic spines. Conversely, prolonged LTD doesn't just remove receptors. It'll eventually destroy the PSD95 egg carton slots entirely, leading to the physical retraction and loss of the synapse. So yeah, your brain is quite literally rewiring its physical shape right now as you listen to this explainer.
But hang on, thinking about all this growth brings up a massive architectural question. If synapses are constantly strengthening every single time we learn something, why doesn't the brain's electrical system completely short-circuit or just max out entirely?
Which perfectly brings us to part 4, synaptic homeostasis and balance.
To prevent runaway excitation or the complete loss of our stored memories, the brain enforces some incredibly strict building codes. The first mechanism is metaplasticity. This basically means that the rules of plasticity actually change based on the history of neural activity. If a neuron has been highly active recently, the modification threshold for future LTP slides up. The brain often achieves this by changing out the subunits of those NMDA receptors, swapping combinations like NR2A and NR2B. The result? It becomes much harder to induce LTP and much easier to induce LTD, which brilliantly keeps the system from maxing out.
Now, while metaplasticity changes the future rules for individual synapses, another mechanism called synaptic scaling adjusts the whole cell right now. Imagine turning down the master volume on a mixing board without changing the individual sliders. Synaptic scaling multiplies or divides all of a neuron's synaptic weights by the exact same factor. This keeps the neuron's overall firing rate totally stable, while brilliantly preserving the relative differences between individual synapses. The integrity of your stored memories stays completely intact.
And finally, part 5, making memories permanent.
Let's zoom back out for a second. How do these temporary molecular tweaks set like concrete into long-term memory? Because initially, these synaptic changes are entirely temporary. Since proteins naturally degrade and turn over, memory consolidation is required to convert short-term adjustments into permanent architecture. This requires molecular switches that stay on long after the initial stimulus, like autophosphorylating CAMK2 and protein kinase MZ. But for true, lasting long-term storage, the neuron must manufacture brand new structural proteins. This requires new gene expression, regulated deep in the nucleus by a transcription factor called CREB.
And this leads us to the super cool phenomenon of synaptic tagging and capture. Imagine you experience something trivial, like noticing the color of a passing car. This weak stimulation just tags the synapse using kinases. Normally, it fades away. But if a major, highly emotional event happens within about 2 hours, that strong event triggers a massive wave of new protein synthesis via CREB. That weakly tagged synapse can then capture those newly synthesized proteins to permanently lock in its own structural changes. This explains exactly why you can often remember seemingly insignificant details that happened right before a major life event.
As we wrap up this explainer, I want to leave you with this pretty provocative thought about the ever-shifting architecture of your own mind. We've seen that our memories are built on fluctuating floods of calcium, kinases, adding and removing receptors, and spines physically growing and retracting. Knowing that the biological substrate of your memories is in a state of constant, dynamic turnover, how much of you is truly permanent? It's a pretty wild question to ponder as your brain continues to rewire itself right now. Thanks for joining me on this deep dive into synaptic plasticity.